Doctors Longo and Roberts discuss the current status of gene therapies in rare neuromuscular disorders in this 8-part podcast series. This is derived from the symposium that was presented at World Symposium 2025 in San Diego, California on February 4 through 7, 2025, and is intended for healthcare professionals only. This podcast includes information about investigational compounds that do not yet have a regulatory approval or authorization for a specific indication. The safety and efficacy of the agents under investigation have not been established, and contents of this podcast shall not be used in any manner to directly or indirectly promote or sell the product for unapproved uses. The views, thoughts, and opinions expressed in this presentation belong solely to the author and are subject to change without notice. The contents of this presentation do not constitute an endorsement of any product or indication by Astellas. In this part, Dr. Roberts will discuss vectors, different strategies, modes of administration, and targets in gene replacement therapies.
Now, in the broader sense, gene replacement therapy seeks to actually deliver genetic material directly into the host cell to influence gene expression. In the most simple idea, one of course has a vector. This is most commonly, but not exclusively, a virus, which can then be given intravenously, for example, and can hope to potentially correct the condition within the individual cells using novel transgenes. And suitable candidate conditions for this, as examples of genetic conditions, are now well understood. And crucially, this applies not only to autosomal recessive, but dominant and even X-linked conditions. And across the piece, one can see, for example, mitochondrial problems, spinal muscular atrophy, as is well known, X-linked myotubular myopathy, Duchenne muscular dystrophy, a very common condition affecting 1 in 3,000 male individuals, Pompe disease, of course, an important focus of the meeting here, but other very common conditions, for example, cystic fibrosis, immunological conditions, and perhaps obviously very crucial in early work on gene therapy, hemophilia. So let's now think about the approaches to gene therapy. One can seek to work at the DNA level and gene replacement. So in essence, one is trying to put a new transgene through into the nucleus that will ultimately be transcribed and translated and produce the important functional protein that is lost. Gene editing, which is a very exciting new technology, or CRISPR technology, actually seeks to modify in vivo the actual mutations that are responsible for the pathogenic production of abnormal proteins and correcting these and actually producing a more normalized protein. But of course, there are also RNA approaches where one seeks to actually repair the mRNA transcripts copied from the mutated gene. So, for example, this may be a novel approach that could be extremely useful in myotonic dystrophy, a multi-system condition. And when we talk about the viral vectors, predominantly we're talking about viruses, those such as adenoviruses and AAV viruses, which have the virtue of not integrating into the host genome, or at least not in a large amount, and those which deliberately seek to integrate into host genomes, such as retroviral or lentiviral systems that may be particularly useful for ex vivo systems. There are, of course, other ways to get genetic payloads into the nucleus, various polymers, nanoparticles, and even cell-penetrating peptides. And nanoparticles in particular is certainly on the ascendant. That being said, in a recent review of the clinical trials in gene therapy, it was certainly the viral vectors that stood out, both in direct gene replacement with lentivirus and AAV, but also actually as delivery systems, for example, for gene editing. An example of what one is seeking to do with AAV, so of course one's seeking to remove the native DNA, insert the new transgene directly into the vector, and of course keen to make sure that there's a high transmission into the capsid, producing a recombinant AAV, which then can be given as a treatment and hopefully produce a therapeutic increase in the functional protein that is deficit in the disorder. How then are we going to get these gene therapies in? And we've also heard allusions to tropism being particularly important. These early pictures will remind you somewhat of the mode of action of enzyme replacement therapy. So we need good cell surface receptors that bind to the viral vector in situ, and these are typically, again, glycosylated receptors. The vector is then taken up into the endosome. It's broken down in the endosome and does not go through the autophagic system and into the lysosome per se. but permeates through the nuclear pore and uncoats, and crucially produces an episome, the hope therefore being of non-integration into the host DNA. Of course, there's then mRNA transcription, and ultimately from transcription and moving out into the cytoplasm, translation and producing a functional protein. And crucially, this functional protein is produced within the cell, so this really becomes an endogenous treatment. If we compare and contrast the two main methods of gene replacement therapy, the in vivo approach gives the vector directly into the patient through the intravenous circulation. And the hope is, and indeed many models are showing this leads to a transduction of a long-acting cell type in which integration within the nuclear DNA per se is not actually required. And typically the vector, of course, is AAV. But another approach is the ex vivo approach, where in essence cells are removed from the patient. They are then transfected with the lentivirus typically, and this produces a transgene within these cells, produced externally, and then reinfused into the patient, but importantly with immunological treatment to prevent any rejection of these cells, and potentially integration into a stem cell system that ensures again a durability of response. Turning to the in vivo approaches, in essence the replacement treatment can be administered by multiple routes. The systemic administration has the major advantage of targeting multiple different systems, but it does of course require a large vector load, and these are expensive to generate. There's obviously a risk of immunogenicity and potentially, and hence the need for long-term monitoring of these patients given gene therapies, off-target effects that could not perhaps have been predicted. This can be given, of course, and most commonly is through the intravenous route, but of course, bearing in mind that muscle itself is a highly vascular tissue, one can, in principle, deliver these intramuscularly. But one appreciates that many of the conditions that we're interested in at this meeting are very organ-specific and, for example, CNS-specific in their issues. So, albeit more invasively, one can target the treatments. For example, giving it directly into a ventricular system to tackle CNS disease, giving it into the eye to tackle retinopathy, giving it even into the acoustic nerve to tackle deafness, for example, in mitochondrial diseases. And although more challenging, it can, of course, be given directly through into the heart, albeit with huge trepidation and excellent interventional cardiologists. Pulmonary delivery systems can be really useful, and again, a little bit like the inhalers we use in asthma, directly targeting the main tissue involvement. And when we think about how we might want to design a gene therapy, we obviously need to be thinking whether we want to be targeting the liver, either as a site of production or as a site of disease, the muscle itself, or of course the central nervous system. And to do this, of course, we need to deliberately develop viral vectors that show great tropism for those individual tissue cells, but also crucially to be thinking about unique promoters that are available in those tissues, for example, muscle promoters, to ensure that one gets full delivery and indeed, more importantly, transcription and translation then into the product in question. So using AAV, one really can try and target very specific systems, and tissue-specific promoters are being developed. And this, of course, takes a lot of work and underlies why it can take so long in the preclinical work to develop these viral vectors.
In the next part, Dr. Roberts will discuss immune responses and other safety concerns related to gene therapies.